I'm Kate Grabowski. I'm an associate professor at Johns Hopkins. I'm a molecular epidemiologist, so a little different than I think some of the folks in the room here in terms of the perspective that I bring. But it's my second year being on the committee, and we're super excited about the lineup we have this week. And thanks for coming, everybody.
Great. I'm Katrina Lithgow. I'm Associate Professor in Oxford, and our research in our group is very much on viral evolution, very much within host and between host scales and linking those scales. And this will be my last year on the committee, which is really quite sad. So thank you very much, everyone, for coming.
All right. And Christian Happy can't join us in person, but he's also a committee member. I think he might be online, but we don't really know if he's there, but he might be. But,
hello, Christian.
Hi, Christian. If you're there, Christian, hello. Right, okay, so, I'll start introducing the meeting, I suppose. So my name's Nick Lohman. I'm a professor of microbial genomics and bioinformatics at the University of Birmingham. This is my first year on the committee of VGE. I think this was like my consolation prize for being booted off the organisation committee for ABPHM, another very good meeting which you should go to. It's a sort of like all pathogens version of VGE, I suppose. And my contribution to this meeting is to add bioinformatics to the title, because I feel like bioinformatics should have some more respect generally, and actually that was a feature of ABPHM that people liked, that bioinformatics was in the title. So that's my small contribution. This is the fifth meeting. It is definitely the fifth meeting, even though it feels like it's been going longer than that. It's every other year, and we had only had a virtual meeting in 2021, we think, which should have been the 2020 meeting. But I think this is a great meeting. It couldn't be more relevant, certainly in the aftermath of the pandemic we've lived through. Certainly, this program, I suppose, has evolved to take in both fundamental research into virus evolution and the methods underpinning exploration of viral evolution, phylogenetics, phylodynamics, bioinformatics, etc. But it's definitely got an applied focus, new methods, case studies, and using viral genomic surveillance to understand outbreaks, epidemics, and pandemics. And it's taken on a bit more of a policy type translation into policy role, with more and more talks. Certainly our two keynotes will both touch on how viral genomic surveillance is really critical for managing and responding to infectious disease threats. And despite the news out of the US, viral evolution will continue going forward. So it couldn't be more relevant in the current kind of political context, with many threats circulating. Mpox an active concern right now, H5N1 very much on the radar, and other viruses, and of course COVID, I think, will also come up many times during this talk, during this meeting. Okay, so I think I've kind of covered the sessions. And we'll get into the first keynote speaker talk. So if you're eagle-eyed, you might have noticed we've had a swap of the keynotes. So the first keynote will be from Tanja Stadler, who is a professor at the Swiss Federal Institute of Technology in Basel. And many of you know Tanja from her fundamental phylogenetic papers and method development that she's done over the years. But in common with many people at the meeting, she's gone into more of a policy and more of an advisory role to the Swiss government, in this case, advising on COVID-19, and that will really be the flavour of her talk, translating our fundamental understanding of viral evolution and pathogen sequencing into methods that can inform public health, both past, present and future. So thank you for coming, thank you for agreeing to swap at short notice, and over to you, Tanja.
Thank you very much, Nick, for the introductory words, and thanks for all the organizers for having us all here, inviting all of us here. It's great to be here. As Nick was saying, our group, our lab, is working mainly on statistical computational methods to analyze genomic sequencing data. And today I want to talk about how we use those methods to actually get epidemiological evolutionary insights and how and when those insights were relevant also for policy makers. So we start with the basis of life. All life is characterized by the genetic code, as much are also the viruses. And what we aim in the group is to leveraging on diversity of those virus sequences towards a better understanding of the evolution and epidemiology of those studied viruses. And so I'll first delve into how we quantify pathogen evolution and epidemiology based on patient samples. So when we have swabs from patients, and from those swabs, you then sequence the virus material. And what can you learn from that? And following that, I'll have some words on pathogen sequencing data sharing, some efforts we were doing in that direction to support the community to share their sequencing data. And then towards the end, I'll go into how some thoughts from our side on genomic surveillance and epidemiology going forward, and some summary and outlook. All right, in epidemiology, a key aspect to know is who infected who, the transmission history. And here I show you a transmission tree. Hypothetically, horizontal lines would be an infected individual, and this individual, say, transmitted at some stage to a second individual, a third individual, so always the horizontal lines being infected individuals. Of course, typically, we do not know who infected whom. We only observe that people got sick. And now I want to show you how the genomes can actually allow us to estimate this past transmission history. So suppose this first infected individual was infected by some pathogen, say a virus, of very simplistic 8 nucleotides, so we can actually read off what's happening. And then the virus proliferates and occasionally actually mutates, and those mutations fixate. And so, for example, this first person, while having started with this infection, the force position changed from a C to a G, and equivalently for all those other patients. And so now when we go out and see infected patients, say 4.5 years ago when SARS-CoV-2 started spreading, we can swab patients and then sequence the material, and that's the information, those are the sequence we would get. And I guess from this graph, you appreciate that if two sequences are more closely together, they more likely share the same or a similar path in the transmission history, because there was little time to actually change, while if two sequences are very different, most likely there was a long time, so many intermediate transmissions between two individuals. And so then in phylogenetics, we leverage on the concept that in this transmission chain, or we estimate the phylogenetic tree being a proxy for the transmission chain, we cluster together sequences which are similar. So we would go, based on similarity, from the sequences to the phylogenetic trees. And in my group, we want to go one step further. We not just want to look at the trees, but we want to understand the underlying dynamics. Say one key underlying parameter defining the dynamics is the reproductive number, meaning for an epidemic, how many people does one infected individual on average infect? If that is 2, one person infects 2, those 2 each 2, meaning 4, 8, 16, so we have very fast exponential spread. And in fact, if we were able to have all the patients of an outbreak being sequenced and reconstruct the whole tree, we could essentially count, how many descendants has the first individual here 2, the second one 1, etc. So we can count the average number of secondary infections. However, from the sequences, actually we don't know who infected whom. It may be this person infects the upper person or vice versa. We just know they share a common ancestor. Plus, typically, we only sequence a small, small fraction of all the individuals. So instead of counting, we actually need statistical tools to integrate over very sparse sampling and thus yet infer how many secondary infections does one infected individual cause. So along one line in the phylogenetic tree, how many offsprings do I get on average? And such such computational methods are also called phylodynamic methods, and they allow us, in fact, to estimate, say, the reproductive number. And the underlying models often used is either a so-called birth-death model, where a birth event is a transmission event and a death event is a recovery event. And so you have a rate for birth and a rate for death, and you aim at estimating the birth and death rates from the sequence of branching events in the tree. Or alternatively, there's the coalescent framework, where you define your population model, so that could be an SIR model or an exponential growth, and then the lineages in a tree coalesce within this defined population size dynamic, and you actually estimate the dynamic. Again, what is the rate of exponential growth? And so at the end, then, from the tree might only be an intermediate, you actually get an estimate of those parameters defining or characterizing the dynamic of an outbreak or an epidemic. For today, I decided not to go into those methods, the birth-death or coalescent methods, in detail, but if you want to chat about that, I'm always more than happy to chat about those methods. That's one of the courses we're doing in our research group. However, I would like to point you to a book we just published, Decoding Genomes from Sequences to Phylodynamics, which essentially describes this whole process from the sequencing of the swabs, or if you're interested in macroevolution of species, all the way to estimating, say, the reproductive number. You find a PDF, it's all for free, so we self-publish in a PDF, so we have all the rights, and we are very happy to share the PDF, and we want everyone who is interested to have a look. It's decodinggenomes.org. You find the PDF, you can download it. If you want a print version, you can also go there and buy a printed copy of the book. And for today, I will use kind of methods described here and what I just summarized in a sketch early on and show you how we applied that to different types of virus data. And actually, in 2016, we were heavily digging into methods, birth-death methods, coalescent methods to quantify epidemiological parameters. At the same time, we decided we actually want to understand better influenza transmission with a focus of influenza in a city. To what extent is it in a city driven by introductions from, say, abroad versus ongoing transmission chains in an, say, average city? As an average city, we took the city where our department is based, Basel, Switzerland. It's 200,000 people big. It has a smallish sized international airport, so it is connected, but also not, Paris or London. And so we asked patients who showed up with respiratory symptoms if they're happy to be tested for influenza and if they would be happy to participate in the study, and many weren't. So we collected more than 500 samples, could enroll patients, sequenced them, and here in red you see the basal sequences falling within this blue-greenish colored global tree. And what you can read off here is, I guess, that pretty much if we sequence in Basel, we sequence the world. It's very connected, and we pretty much find every, call it variant, subtype, whatnot, which is globally available. We also find it in Basel. And so that already highlighted that travel and international connectivity, unsurprisingly, I guess, but plays a major role in the spread of pathogens, making us also at the start of the pandemic a bit skeptical on how effective travel bans will be, because it seems it's around the globe before we really worry about it. The actual motivation for this study, however, was to identify Swiss ongoing transmission chains, so Basel transmission chains, looking at the small Swiss subtrees. So, if red dots cluster with within a small subtree, look at that tree and estimate the reproductive number for all those Swiss trees to get an overview. What is the reproductive number of influenza for that particular season? And for influenza, one knows, I mean, it's above 1, otherwise we wouldn't see outbreaks, epidemics, but maybe between 1.1 and 1.3, depending on the season. The fact that most infections were actually introductions rather than through a within Basel transmission event, we had very small ongoing transmission chains, meaning also the uncertainty in the reproductive number was quite wide, and it was, in December a bit higher and then went down. I mean, I think you don't need a big study to say it's somewhere above 1, but below 1.5. So the insight retrospective was more on how much we get from abroad, and the details on local transmission chains we couldn't disentangle as much because there was too little local transmission. It was so much coming in from abroad. So well, then discussing, should we do the study bigger, that we can study a bit more in detail the Swiss local transmission chains. The situation evolved, and early 2020, we had a pathogen rapidly spreading, SARS-CoV-2. And at the beginning of the outbreak, there were a lot of discussions what actually the reproductive number is. Here I call it R0, so before any interventions took place, and while everybody was still susceptible, what was the reproductive number? And remember, for influenza, 1.1 to 1.3 in a typical season. Colleagues of us in Bern, they used some data from China, confirmed case data, and saw, first there were, say, 20 cases, and roughly a week later, 40 cases, 80 cases, and assuming every week, roughly, you infect the next generation of infected individuals, the reproductive number would be around 2. It doubles every week. That's all based on confirmed case data. There, of course, you have to rely on that the reporting of that data is reliable. We had then the thought of, we do an alternative study to, see if we can confirm this number. And namely, we used the first sequences which were being published from around the world and estimate for the different locations the reproductive number. And so, of course, China had the first cases and first sequences. Then also in Europe, Italy, we had some sequences being available, Washington State and the US. And those 3 countries showed, using those phylogenetic tools, so we used genomics sequencing data, reconstructed the phylogenetic tree, and then asked, what is the average number of secondary infections, using statistical tools to average out that we have a huge sparse undersampling. We got a reproductive number of somewhere, a bit higher than 2, but within its uncertainty, probably more towards the direction of 3. The Diamond Princess, you may remember, was a cruise ship. They had one of the first outbreaks. Here, the reproductive number was a bit higher in our estimates. I guess it makes sense. It's a very closed environment. People have a lot of contacts. And the prior, I should say, it was all done with Bayesian methods, was, a bit lower. So there seemed to be clear signal. It's somewhere, all studies taken together between 2 and 3. And that, based on epidemiological models, means that more than half of the population will get infected before the peak of the outbreak is even reached. And after the peak, of course, you can get close to the same number of infections again. So highlighting that the world is facing a huge problem because infections are so severe, and the vast majority of people will get infected if no interventions are being taken. All that was done before, say, Switzerland, for example, even had the first SARS-CoV-2 case. And at that time, though, based on those numbers and what many colleagues around the globe were doing, I guess we were all convinced there'll be a huge problem, many infections. So we were exploring ways to sequence SARS-CoV-2 in Switzerland when it will start spreading. And so we teamed up actually with a diagnostic company. They were in Switzerland. People know about it because, if you go to the doctor and they do any test, it's very likely you get from them a bill. They are a very big player there. And they were scaling up to be prepared to do the PCR testing on large scales in Switzerland once we have more cases. And so they agreed to share with us the swabs, which were done for the SARS-CoV-2 testing, PCR testing, and we would then sequence them, so the virus. And if you only target in a sequencing and the bioinformatics the virus, the ethics situation is that you can use that without the patient consent, because at the end you only have the virus. And so this company, actually we started then in spring 2020 on a weekly basis. They shipped to us samples, when they were very overwhelmed with PCR testing. Some of us went over there and put together the boxes on ice to get the samples and ship them back, which was also a way for some of the team members to get out of the lockdown and go to their lab and get the samples together. And so on a weekly basis, we did roughly 1 plate of SARS-CoV-2 sequencing. And this is the tree here shown through a next-strain view of the first year of the pandemic. The dots would be the Swiss sequences and the lines, the global context. And so what can we now learn from the tree? I made the point you can look at many things and look also at growing subclades, but what can we do regarding quantification? So based on this tree, Sarah Nadeau, who was a PhD student at the time in the lab, applied then those methods on estimating the reproductive number, assuming it can change on a weekly basis through time, and making different assumptions what is actually a Swiss cluster. So from all the Swiss cases, we can't build one tree, because we have, as I made the point earlier, many introductions from abroad. So we used, and there also thanks to the UK, we had a lot of context sequences. So we carved out Swiss transmission clusters based on the rich diversity of and amount of UK sequences. And then based on how we defined what a Swiss cluster is, we had 2 different kind of estimates of the reproductive number. And clearly, the first wave, it was high, and then here's the second wave, and the pattern broadly agrees based on when we estimated the reproductive number based on confirmed case data. Here you could now say, though, why would you do it on sequences if you have reliable confirmed case data? And I would agree, that's good for confirmation, but it doesn't really lead an added value, in particular because the uncertainty is much bigger here, since the amount of data, the amount of sequences typically is smaller. However, we wanted to assess if contact tracing in the first year of the pandemic actually had an effect on transmission rates. And we were thinking, you can try to do correlates. Does transmission go down once you have contact tracing working well or not? And this is all very well. We thought, can we also go an alternative way and look at the sequencing data in the way of saying, we are identifying those Swiss subtrees in a global context tree and say those are 2 Swiss subtrees. And now, after an introduction, contact tracing can only kick in once we know about the first case of this cluster. If we don't know that somebody traveled, say, from the UK to Switzerland, who is positive, and this positive case is identified, contact tracing will not start, because they only, of course, can start once they know about a case in a cluster. And so we were saying, and that's of course simplistic, we know about this transmission cluster once we know about or have the swab of a later first identified positive case in a cluster. Of course, not everybody gets sequenced, so there may be a confirmed case already earlier, and then contact tracing kicks in. But of course, also contact tracing doesn't start right on the day you're testing positive. At least in Switzerland, it took a while until everything, everybody was informed. So we said this is just a proxy. The first sequence is when transmission potentially slowed down in such a cluster due to contact tracing. And so we then use this burst death model. So you have transmission and recovery. And we say transmission up to the first case in a cluster, be it here or in this cluster, it's the first 3 transmission event, and this is the first sequence then. Up to here, it is this, background reproductive number, and then you have some dampening factor. So by some factor R, transmission may slow down once we know about the first case. And within the phylogenetic framework, we use BIS2 for that. We implemented that model and actually could estimate that in summer 2020, when in Switzerland the cases were actually low and contact tracing was started, depending on the precise assumption, roughly half of or the transmission rate was roughly halved upon identification of the first case. While then in fall, when we had a really big second wave and it was reported that contact tracing was fully overwhelmed, also here the dampening parameter got down close to 0. So it seemed to have, transmission was not being slowed down after the first sequence was identified. In spring, one needs to say we don't have very conclusive results, but I guess everything is rather uncertain, so there needs to be more data. There's not enough data early on to make conclusions there. I would highlight that here, what we really estimate is how much the transmission slowed down after you or after the first case is identified. We say it's plausible that this is connected due to contact tracing, but it could, of course, the causal relationship we can't prove. We just pretty much exclude other scenarios. We couldn't come up with another good explanation why things would go slower. So this was kind of a proof of concept that actually contact tracing can work. And of course, there were many other studies that it can work if it's done efficiently and fast, in particular in times of low case numbers. And for Switzerland, we could quantify the effect. So, and in the quantification, well, as I said, spring and fall, spring is not, it's not informative, the data. For fall, there's relatively little to no impact. In summer, it was 35 to 63% of slowing down. So if before you had roughly a reproductive number of 2, if it's 50%, you can actually bring it to 1 or just under 1 and control the outbreak. So it seemed to have been quite efficient for, admittedly, a few weeks. Okay. The other part of the story was why we started sequencing was, of course, we had in mind we can quantify reproductive numbers and evaluate public health interventions, but really we want to understand, while we know the virus changes all the time and we leverage on diversity, so mutations appearing, we can construct those phylogenetic trees. Thus far, we looked at everything as being kind of neutral. All the viruses are having the same properties. One big question, though, was, and is also for any other virus, are certain variants evolving, certain mutations coming up, which really changes the phenotype of the virus. And for SARS-CoV-2, the WHO defined those variants of concerns, where what we should look at, meaning either a variant has an increased transmissibility, causes more severe infections, or evades immunity acquired through previous vaccination or infection. And what we, as well as, I guess, the global community set out to look in the sequencing data, do we see any signs of features popping up in that direction to have an early warning? And in fact, in summer 2020, Emma Hotcroft, who is also in the audience, she looked in detail at the sequencing data and saw some variant just growing in the tree. And she works in the next strain team, and they saw in their tree there is this, I call it for now, orange variant. This cluster is just increasing rather fast. And so she then digged deeper, and we looked across Europe. And in particular, I mean, the UK also was pretty much the only variant, orange. In the UK, Spain, it was completely dominant. And other countries, also Ireland, it increased in frequency. And so we were, the first time we saw it, this is now more retrospective, we were a bit, alerted that it could point to a transmission advantage. And we also informed our public health agency that there is a new variant. We're not yet sure, we're studying, but to give a heads up that we are closely aligned in case additional things happen. Then Emma digged in further and actually realized or found out that in Spain, this orange variant first appeared among agricultural workers and was then connected with some super spreading events. So it was a fully epidemiological type of information. And those super spreading events seem to have reseeded the epidemic after the lockdown numbers were very low. And then it was, just the orange got a head start to super spreading, was spreading. And then our hypothesis was that in particular from Switzerland and the UK, there was a lot of summer travel in 2020 to Spain, and that was possible under the rules. And those people, of course, then, if they get the virus in Spain, coming back, if you travel to Spain for summer vacations, you're probably also having some social contacts back in your home country. So the orange strain might have just been in a more mobile population and had an advantage due to demographic factors, but not due to the virus. And in fact, this then was more epi data, was more our working hypotheses. And then retrospective, we had a second wave in fall and all over the globe. We estimated then the reproductive number of this new variant, and here it actually doesn't show orange, now it's all of a sudden red, but this is also more technically called 20A.EU1. Actually, the reproductive number was roughly the same throughout the second wave. In particular, it's not that orange or this 20A.EU1 caused the second wave. The other variants roughly had the same dynamic. So really, it was a host effect and not a virus effect. I tell you that story because I think it highlights that with the sequences, we can go a very far way. And it also raises, gives rise to hypotheses. But we should always, if there's a public health consequence, carefully check back with other epi data before we draw too strong conclusions, which might be wrong. However, it was a very dynamic time. We didn't have to wait long for the actual variant of concern to appear. And here I first want to say that in that time we looked more and more through the data. As I said, Emma was kind of looking at the next strain tree, and we did a lot by hand. I was also going through the data. And then Tara and Chen joined our team and with the task to look and identify or quantify evolutionary dynamics of SARS-CoV-2. And he set up this dashboard over the years, and it really started at the time when the first variant of concern arrived, what is now called cough spectrum, where all the 15 million or by now 18 million of available SARS-CoV-2 genomes you can query. They're in a database. You can see how the different variants in your country of origin or country of interest, what the status is. But you're not fixed to a particular named variant. You can query for any arbitrary combination of mutations. You want to see, is there some strain with mutation A but not B and maybe C because you think that has an immunity effect, etc. You can query that. You get the answer interactively in real time. That was at this, it sounds like, what else on a dashboard? Of course, you should get things in real time. The problem was, at the time, the number of sequences was growing so rapidly, and you can compress sequencing data, but if it's compressed, you can't query it. And so Charan, together with colleagues in a group, then developed a smart way to compress on a column-based way the whole alignment such that it is still queryable for mutations. And now, with those over 15 million sequences, you get within milliseconds your answers. We used, I should say, if you want to find out more, Charan is in the audience. And tomorrow, there's the poster 8 and the pitch demo 8. I all encourage you to go there and find more out of cough spectrum. And then more generally, he's generalizing that at the moment to other pathogens. So go there and hear the latest developments. We used or developed those approaches, in particular, once the first variant of concern was characterized, alpha. It was reported from here in the UK. Was end of 2020. Case numbers were going down in Switzerland and many places, actually. And then upon the UK alerting us about alpha, we looked in the data and we saw, indeed, it's still low, but it's a bit increasing. And if you then fit mathematical models, logistic growth models, so very simple, 2 strains competing, it seemed there's a transmission advantage of 40 to 50%. That was estimated, not particular, we estimated for Switzerland, but around the world, you get the same numbers, unsurprising. It's an intrinsic virus advantage. And we anticipated in March there'll be another wave. And actually, our government at that time, we were in the communication between science and the government after a first year of it being a bit bumpy. It improved quite a bit. We're in close contact, and they actually strengthened measures when case numbers were dropping quite a bit, because they anticipate what the models were saying, that alpha will become dominant and that we will not circumvent, because that's just a competition of two strains. But what we can do with measures to dampen the overall wave. And so they strengthened measures, which was a bit of a communicative challenge, I suppose, because the general public didn't understand why do we have stronger measures now that it's going down compared to when it was going up much earlier. So those are all screenshots from cough spectrum, and I invite you to, play around if you're interested in SARS-CoV-2 variants yourself on the dashboard. The link is down here, coughspectrum.org. With that, I want to switch gears a bit and go to the second part of the talk, thoughts about pathogen sequence data sharing. So as I said, we developed for cough spectrum infrastructure that we can host large amounts of sequencing data, and that we can also, through an API, interactively query it. And so then we teamed up with people around the globe, in particular Emma Hotcroft and her team. Emma, I already mentioned, is here. Theo Sanderson was also somewhere in the audience, Charan, who was mentioned, and in particular Artem Babaya from the University of Toronto, and Richard Neer's group, and software development support, to actually leverage on all those infrastructure developments we did for particular projects, dashboards, that we can offer open source software to share genomic sequencing data. It's called Localus. It is alive. You can download it, try it out, localus.org. And in particular, it is a software you can host your own database. People, if you host that, can submit data. The processing goes through the Nextclade pipelines. Probably NextStrain, and Nextclade is from the same team doing the processing of data. You can submit and access data through an API. It's a fast API called Lapis, which we actually developed for the cough spectrum project. And as said, the real kind of added benefit is this smart compression with Charan and Alex Tepper from the lab developed to query large amounts of data, which is compressed. And so, if you want, you can set up your own genomic sequencing database. For example, if you work on HIV and your data is not being able to be shared, but you want to share it within the lab, etc., we envision or have the vision that over time it gets to a tool which people can easily set up and use, compared to the pandemic, where across the world, many of, including us, we set up our, on-the-fly kind of sharing platforms within labs, within countries, etc. And it's early days, it's up and alive, but if you have any feedback or want to contribute, it's all open source, please contact us. And again, there will be a poster on it and a pitch today, later, also by Charan Chen, so I invite all of you to find more about that later. And Localus is already used by a bigger data sharing effort called Pathoplexus. So Pathoplexus was launched a month and a half ago, and currently it supports four types of viruses. So the Korean Congo hemorrhagic fever, Ebola Sudan, Ebola Zaire, and West Nile virus. And essentially, Pathoplexus is a platform like other databases where you can submit your data, you generate it, and you can access then data which is shared by other researchers for further analyses. And the Pathoplexus is based on the Localus software. And in particular, it's run by an international executive board from the community for the community. So while I presented now Localus and we are heavily involved, the running of Pathoplexus is something independent. And here you see the executive board. It's actually 5 researchers from 5 continents. Emma Hotcroft, also representing more, or the deaf side of things, is representing in a way Europe. It's not that only one person per continent may enter, but the idea was having it very diverse. George Ghisini, who should also be somewhere in the audience, He represents kind of the African continent, Alison Black from the US, Anderson Brito from South America, and Tommy Lan from Asia. The core principle on pathoplexis, or in the governance structure, is everyone has access to all the data at all times. So you don't need to, say, sign up or do anything particular. Everybody is guaranteed to have access. Also, programmable access through APIs is always possible. So if you have a dashboard, you can directly feed your data in, and you don't have to go and click through buttons. It's all programmable. We envision that, or you tell us, but that data submission is easy and very straightforward and not as complicated potentially, or too many things to keep in mind as in other databases. And in particular, if you're working on a scientific paper, you can ask there also for a 1-year restricted period where other people are not allowed to publish papers or preprints on your data. However, you would agree if you upload there that people can use it for public health benefits. So if people do public health analysis looking at, say, for SARS-CoV-2 new variants or Mpox at the moment, or once we have an instance for Mpox, public health analysis are encouraged, and as a global community, we feel this should be done no matter who publishes later the data, but the researchers who want to publish have a 1-year kind of period where they can publish. After that, actually, your life is easy because data will be pushed to ENA. You don't have to bother with that anymore. So once you publish in the journal, also the data access, open data is made possible automatically. And your sequences get as sets together a DOI. So if then people want to cite you, they cite a DOI on your sequences. In that way, pathoplexis has the vision that data submitters are better recognized, also quantitatively, for their contributions facilitating both research and public health. I only sketched over that. More will be in Emma Hodkoff's talk tomorrow at 5 to 5. So genomic surveillance and epidemiology going forward. I do think, I talked right now about patient samples and sequencing. I do think this needs to be continued, that we have some information on what's circulating in patients. And I'll very briefly outline some efforts we are currently setting up in Switzerland to do so. But I also do think at the same time, while we need a limited number of patient samples to get a population overview, we don't have to sequence tens of thousands of samples from patients. We can look in environmental samples where there's the average of what's around in a population. And there actually we digged into wastewater as one environmental aspect where you can get averages of populations. And so first, about the patient sequencing, most recently I told you about SARS-CoV-2 swabs coming from this diagnostic company Violier. By now, they actually provide to us samples from patients which tested positive for respiratory, I should say, patients who show up at the doctor with respiratory symptoms. And those swabs, so they will then get tested for some diagnostics, but those swabs we sequence with a broad panel. We currently use the Illumina respiratory virus panel, and then we identify, whatever virus this infection was causing, so we get a broad overview of what's circulating. Here, this is all Swiss samples. Charlene, who is also in the audience, as part of her PhD, set up a great dashboard to interactively look at this data, also from a geographic perspective. That's Switzerland with its 26 cantons we have. And you can also explore the quality of the genome's coverage, etc. And Charlene has a poster on that and a pitch also today. So now another advertisement, inviting you to go to that pitch. We only did one season so far. We have, less than 100 of sequences for some of the viruses we looked more closely into. It doesn't look that impressive, but compared to what's available on NCBI, so openly available for Switzerland, we have one RSV genome and one HPIV genome. So that's all there is. So I guess Charlene's pilot already adds a lot of data. Globally, there's not that much either. And our vision is, in particular with this panel, to investigate RSV in more detail as monoclonal antibodies are being rolled out, the vaccines are rolled out, so what happens there evolutionary. And as we saw for influenza, if we sequence in Switzerland, we pretty much sequence global diversity. I anticipate the same for those viruses. The seasonal coronaviruses are not studied very much. I mean, also for reasons they don't cause too severe infections, but I do think it would be very interesting to understand those 4 seasonal coronaviruses, what their dynamics are, is their inhibition, etc. Also, because now we have a 5th coronavirus still much more virulent, but another coronavirus and HPIV. We're focusing on going forward, and we want to sequence at least for another 2 years, and then, of course, hopefully more to come in one of the next conferences. For the community to get averages, you can actually look in wastewater. And this really accelerated that field during the pandemic. Here on an hourly basis, automatically wastewater from the ETH Zürich plant is being collected. And then overall for 14 sites in Switzerland, that's being done, sent to the lab, sequenced, and PCR quantification is done with digital droplet PCR. And then essentially you can, you see what's circulating, and it's routinely done currently for SARS-CoV-2, influenza, and RSV. And the labs are in the process of doing pilots for mpox and seasonal coronaviruses, where again, I feel the seasonal coronavirus, we have the opportunity to really also understand in summer, how much is it there? And here to close now the loop, this is digital droplet PCR data. We see, a wave of SARS-CoV-2, and then Adrien Lison developed a tool to translate that or estimate from that the reproductive numbers through time, so one can use that for surveillance purposes. And we do it now for the 3 different viruses we monitor. And on the dashboard wise.ethz.ch, you can actually here see updated as soon as new data comes in what the current reproductive numbers are. To close the whole loop, though, I don't want to make the impression we're focusing everything on surveillance. I think from doing good surveillance, you have tons of data to actually understand epidemiology better. One example here is where Sarah Nadeau comes in again. She was the contact tracing, leading the contact tracing study. Looking at influenza, when just in this year when Omicron appeared. And so we didn't have very strong measures in Switzerland when Omicron arrived, and then measures were introduced, so the black dashed line. Influenza in blue in wastewater crashed, and upon release of measures, we see a wave again. And to some extent, in confirmed cases, we see it depending on a location a little bit, but I believe with wastewater, we have a way cheaper tool to more broadly get those blue curves, so this is way fewer sample points than the red one, and understand the dynamics and be able to quantify how different interventions actually, what effect they have. And with that, I'm at the summary and outlook, so I hope that you saw how sequencing and statistics actually can lead to real-time insights into pandemic spread, and actually for Switzerland, the first year was not very good, the communication then to the policymakers. I mean, not very good. There was not much communication. What should be done with research insights, but then it got better. So I think there is opportunities to continue such genomic surveillance efforts and institutionalizing those policy interactions, because there's many new pathogens threatening us at the moment as we speak. I guess challenges will just be, will countries keep up monitoring pathogens? For Switzerland, unfortunately, more and more of that moves. It was also always partially funded by research, but also partially funded by public health. It more and more goes back to the research side, but there also you don't get money for surveillance, which also it shouldn't. It's not, you do the research and do prototypes, and then whatever should be surveyed needs to come from public health. Timely data sharing, it was not always easy throughout the pandemic, and I think we need more discussions in the community. How do we through databases, share the data and ensure nobody's scooped and everybody's recognized, and also public health has the relevant data at all times. And of course, generally, globally, how do we talk effectively with politicians? And with that, I'm at my phylogenetic tree of acknowledgements of all the people involved and all our funders, and I'm looking forward to your questions and discussions over the next 2 days. Thank you.
Fantastic. That was a brilliant way to kick off the conference. Thank you, Tanja. We have a couple of minutes for questions, so it'd be nice to hear some from the audience. There's microphone runners all around. Anyone like to start? Do you want me to invent one? Rachel, there's a question here.
Hi. Yeah. So you, in your presentation, included several of the lovely dashboards of the data from Switzerland, the surveillance data from Switzerland. Are they all completely open access? Is the data behind that available for anyone?
So for the wastewater data, all the data is available. And for our, I should say, everywhere where we show our own data, it's either available. Charlene is about to push everything to GenBank, so it might, depending how lately that was, but it's all available up on as we speak. If it's global dashboards, and that's in particular the cough spectrum dashboard, there's 2 versions. There's an open version based on GenBank data, INSDC data. Then, of course, you can just go on INSDC. And there's a GIS-8 version. And then, depending on your access to GIS-8, you can access the data or not.
Amazing, thank you.
Another question. I'm going to ask. Oh, there's a question. Oh, yeah, here we are. There's a hand. Very good.
Thanks, Tanja, for this talk. Very interesting. I was wondering regarding all the work you are doing to monitor pathogen circulation. Do you know if this will be used later as I don't know, early detection warning signal or something like that? Like, what the next step after this? Thank you.
So, for the wastewater, and I should say we are more on the computational and modeling side, but our collaborators doing the assays for the wastewater, they are working actively on assays where, you can distinguish avian influenza from human influenza, et cetera, et cetera. So there's a lot of work going on with respect to assays, et cetera, and to do that fast. So I would say ongoing work, in fact, indeed, both in Switzerland but also internationally.
Okay. Any questions online? Nothing online. There's like 400 people online, isn't there? Anyway, right. So we're actually at time. I think we should try and keep the session to time, so we'll stop there. And thank you again very much for that excellent talk.